Welcome to BioCrack Academy. In this video, we will cover MCQ question on the microscopy with clear answers, perfect for quick revision and exam preparation. So, let's get started. The first question is, what is the typical magnification range of a stereo microscope? 10x to 100x, 1000x to 200x, 1x to 10x, 10,000x to 1 lakh x. And the correct answer is 10 to 100x. Next question is, which microscopy technique uses a beam of ions? Focused ion beam microscopy, transmission electron microscopy, scanning electron microscopy, or confocal microscopy? And the correct answer is focused ion beam. Next question is, what is the primary use of dark field microscopy? To view internal cell structure, to observe live unstained specimens, to study chemical reactions, to measure specimen thickness. And the correct answer is to observe live unstained specimens. Next question is, which component of the microscope align the light path? Condenser, diaphragm, nose piece, or stage? And the correct answer is condenser. Next question is, which lens provide the initial magnification in a compound microscope? Eyepiece, objective lens, condenser lens, or diaphragm? And the correct answer is objective lens. Next question is, what is the resolution limit of a scanning electron microscope? 0.1 nanometer, 1 nanometer, 10 nanometer, or 100 nanometer? And the correct answer is 1 nanometer. Next question is, which microscope is used to study the optical properties of crystals? Polarizing microscope, fluorescence microscope, confocal microscope, or scanning electron microscope? And the correct answer is polarizing microscope. Next question is, what is the purpose of the objective lens with a shortest working distance? To provide the highest magnification, to illuminate the specimen, to hold the slide, to adjust the focus? And the correct answer is to provide the highest magnification. Next question is, which microscopy technique is used to study super resolution imaging? STED microscopy, phase contrast microscopy, dark field microscopy, or bright field microscopy? And the correct answer is stimulated emission depletion microscopy, or STED microscopy. Next question is, which scientist improved the microscope to observe single-celled organisms? Robert Hooke, Antony van Leeuwenhoek, Hans Janssen, or Louis Pasteur? And the correct answer is Antony van Leeuwenhoek. Next question is, which microscope uses a technique to achieve resolution below 200 nanometer. Optical microscope, super resolution fluorescence microscope, scanning electron microscope, or phase contrast microscope. And the correct answer is super resolution fluorescence microscope. Next question is, what is the primary source of electrons in an electron microscope? Tungsten filament, LED light, laser beam, or X-ray tube? And the correct answer is tungsten filament. Next question is, which microscopy method is used to study the elasticity of the biological samples? Atomic force microscopy, transmission electron microscopy, confocal microscopy, or fluorescence microscopy? And the correct answer is atomic force microscopy. Next question is, what is the function of the beam splitter in a confocal microscope? To magnify the image, to direct light to the detector and specimen, to adjust the stage, to focus the light? And the correct answer is to direct light to the detector and specimen. Next question is, which microscope is the best for studying the internal structure of viruses? Scanning electron microscope, transmission electron microscope, optical microscope, or stereo microscope? And the correct answer is transmission electron microscope. Next question is, what is the purpose of a vacuum pump in electron microscopy? To cool the specimen, to prevent electrons scattering by air molecules, to magnify the image, to adjust the light? And the correct answer is to prevent electron scattering by air molecules. And the next question is, which microscopy technique uses a contrast based on difference in refractive index? Bright field microscopy, phase contrast microscopy, fluorescence microscopy, or dark field microscopy? And the correct answer is phase contrast microscopy. Next question is, what is the typical sample preparation for scanning electron microscopy? Staining with dyes, coating with thin layer of metal, freezing in liquid nitrogen, or mounting without preparation? And the correct answer is coating with a thin layer of metal. Next question is, which microscope is used to study the magnetic properties of thin films? Lorentz transmission electron microscope, confocal microscope, fluorescence microscope, or phase contrast microscope? And the correct answer is Lorentz transmission electron microscope. Next question is, what is the role of the detector in a scanning electron microscope? To magnify the image, to collect secondary and backscattered electrons, to illuminate the specimen, to adjust the focus? And the correct answer is to collect secondary and backscattered electrons. Next question is, which microscopy technique is used to study the 3D structure of thick tissues? Two-photon microscopy, bright field microscopy, dark field microscopy, or phase contrast microscopy? And the correct answer is two-photon microscopy. What is the typical field emission gun used in modern electron microscope? Tungsten filament, cold cathode, Schottky emitter, or LED source? And the correct answer is Schottky emitter. Next question is, which microscope requires the use of a fluorescence probe? Transmission electron microscope, fluorescence microscope, scanning electron microscope, or stereo microscope? And the correct answer is fluorescence microscope. The next question is, what is the purpose of the objective lens with the longest working distance? To provide the highest magnification, to allow observation of the thick specimens, to reduce light intensity, to adjust the stage. And the correct answer is to allow observation of thick specimens. Next question is, which microscopy method is used to study the dynamics of molecular motors? Total internal reflection fluorescence microscopy, scanning electron microscopy, phase contrast microscopy, or dark field microscopy? And the correct answer is total internal reflection fluorescence microscopy. Next question is, what is the main advantage of a digital microscope over a traditional one? Higher magnification, direct image capture and analysis, lower cost, or simpler optics? And the correct answer is direct image capture and analysis. And the next question is, which microscope uses a technique to image non-conductive samples without coating? Environmental scanning electron microscope, transmission electron microscope, confocal microscope, or atomic force microscope? And the correct answer is environmental scanning electron microscope. Next question is, what is the typical magnification of an atomic force microscope? 10 to 100x, 1000 to 10,000x, up to 10 CRX, or 1x to 10x? And the correct answer is up to 10 CRX. Next question is, which microscopy technique is used to study the thickness of thin films? Ellipsometry microscopy, fluorescence microscopy, phase contrast microscopy, or confocal microscopy? And the correct answer is ellipsometry microscopy. Next question is, what is the purpose of the anti-vibration table in microscopy? To magnify the image, to reduce vibration affecting image quality, to adjust the light, to hold the slide? And the correct answer is to reduce vibration affecting image quality. Thank you. Thank you for watching. If you like the video, please share, comment and subscribe. Thank you again.